Good morning, good afternoon, and good evening to the speakers and attendees joining from regions around the world. It is my pleasure to introduce our new questions and answers for ICH E14 and S7B. They focus on an integrated risk assessment and how to use non-clinical data, not just before first-in-human studies, but also to inform clinical decisions later in development and to inform regulatory decisions and labeling at the time of a marketing application. While QT prolongation gets a lot of attention, it's important to note that QT prolongation isn't associated with any patient harm by itself. The real problem is drug-induced torsade des points, or twisting of the points, which I'll refer to as TDP. It was recognized in the 1990s to early 2000s that many drugs outside of cardiac drugs could cause TDP, and sometimes this could lead to sudden cardiac death. So the community came together and asked, what do torsade-causing drugs have in common? And it was recognized that they prolong the QT interval on the electrocardiogram, prolong the action potential at the cellular level, and block the HERG potassium channel in the cell membrane. With this information, ICH guidelines were developed. ICH has a mission to achieve greater harmonization worldwide to ensure safe, effective, and high-quality medicines are developed and registered in the most resource-efficient manner. The process for ICH guideline development involves 5 steps. The first step is to build consensus among experts on an ICH working group. The second step is to achieve consensus and adopt the draft guideline. We have done that with the ICH E14 S7B Q&As and are now in step 3, consulting the public. We will then discuss comments received, revise as needed, and finalize the guideline. Step 4 is to adopt the ICH harmonized guideline, and step 5 is to implement the final guideline in regulatory regions around the world. ICH S7B was established in 2005 and describes the non-clinical cardiac safety pharmacology studies to assess whether a drug blocks the HERG potassium channel, prolongs the QT interval in in vivo studies, and then follow-up studies can be conducted as needed. It has been successful in bringing investigational drugs forward safely into first-in-human studies. ICH E14 was also established in 2005 and describes the clinical guideline describing the so-called human thorough QT or TQT study. It established a very sensitive threshold for ruling out TDP risk. It's approximately a 2% increase in the QT interval. This is very small, and thus it resulted in it being the most intensive and expensive clinical pharmacology study in drug development. There have been multiple prior E14 Q&As to introduce incremental steps forward in the science. Overall, the guideline has been successful in preventing drugs with unknown TDP risk from reaching the market. However, there's room for improvement. The S7B studies currently inform safety before first-in-human dosing, but then are largely ignored, as shown by this red bar between non-clinical and clinical. Clinical assessment then relies on the human QT interval, which is an imperfect biomarker. It's sensitive, but not that specific. Prior E14 Q&As only allow for TQT study substitution, that is with phase 1 concentration QT analysis under narrow requirements. Specifically, very high exposure is required, which we'll come back to later in the talk. The prior E14 Q&As also only allow for limited decision-making when a TQT study or its equivalent in a TQT substitute cannot be performed. It results in unclear risk determination and unclear labeling, such as having no large QT effects. E14 also states that the TQT study is intended to inform whether ECG monitoring is required in phase 3 trials, as not all QT prolonging drugs are proarrhythmic. However, the practice has been that drugs with a positive HERG or QT signal are often dropped from development, and we may be losing promising, effective drugs due to false positive signals. While at adoption, E14 did suggest a QT interval evaluation independent of S7B results, and that's how it has largely been practiced, both documents do highlight the need for integration of information in a manner which is informative as a totality of evidence approach. Our new Q&As are directed at scenarios where the non-clinical data can reduce the number of clinical studies, specifically the thorough QT studies, streamline drug development, inform clinical regulatory decision-making at the time of a marketing application with the non-clinical data, and to inform labeling to better communicate risk. In 2018, the E14 S7B discussion group reached agreement on a two-stage approach to link S7B and E14. Stage 1 focuses on S7B Q&As on integrated risk assessment, best practice considerations for in vitro and in vivo assays, and principles of proarrhythmia models. In addition, Stage 1 focuses on E14 Q&As on how to use the non-clinical data to inform regulatory decision-making in late-stage clinical development and at the time of a marketing application. In the past, the S7B non-clinical data has not been used to do this. This image is taken from the original S7B guideline. It shows how the in vitro IKR assay and the in vivo QT assay are the core non-clinical assays that inform the integrated risk assessment. If needed, relevant non-clinical and clinical information can be taken into account and follow-up studies can be performed. This informs an overall evidence of risk. With our new Q&As, we focus on the integrated risk assessment considerations when non-clinical data are used prior to human testing, as was the primary focus of S7B, and also later in clinical development for E14 scenarios and how we will use the non-clinical data for clinical decision-making and regulatory decision-making and labeling late in development. In addition, the new S7B Q&As focus on best practice considerations for ion channel assays, specifically the HERG assay as part of a core assay, and the in vivo QT assay, the other core assay. The ion channel best practices can also apply to follow-up studies if additional ion channels are assessed. And we have Q&As on the best practice considerations for myocyte assays, including with induced pluripotent stem cell-derived cardiomyocytes, and the principles of proarrhythmia models for risk prediction. There are two scenarios to use non-clinical data to inform clinical decision-making in our new Q&As. The first is the so-called double negative scenario, when there is no HERG block and there is no QT prolongation. The second is the so-called non-double negative scenario, when there is HERG block and or QT prolongation. In the revised E14 Q&A 5.1, we will be able to use double negative non-clinical data to decrease the need for thorough QT studies. Specifically, it will allow for additional TQT substitutes when the drug exposure in concentration QT analysis from phase 1 studies is not high enough to meet the current requirement. What do I mean by that? Well, on the right, we have a pharmacokinetic profile that could be from a TQT study or a phase 1 study. The requirement for a TQT study is to cover the high clinical exposure. However, the requirement for a TQT substitute with concentration response modeling is to achieve 2 times the high clinical exposure. This is harder to achieve, And with the new Q&As, you will be able to qualify for a TQT substitute by only reaching the high clinical exposure if there is supporting non-clinical data. This can reduce the number of clinical studies in drug development, affect a large number of drugs, as approximately a third of QT studies over the past 5 years fall under Q&A 5.1, and only approximately 40% of those cover 2 times the high clinical exposure. In addition, in the revised E14 Q&A 6.1, we can use double negative non-clinical data to inform regulatory decisions and labeling when a TQT study or its equivalent cannot be conducted because of situations like having safety concerns with healthy volunteers. This happens in oncology, but also other therapeutic areas. Feasibility concerns in patients that result in a lack of a positive control or inability to achieve high exposures, and a confounded QT assessment, such as might occur with large heart rate changes. The impact is that we can change regulatory decision-making and labeling, as these cases often result in a finding of no large QT effects with current practice, but with the new Q&A, a conclusion of low risk can be reached with appropriate non-clinical data. This has the potential to affect a large number of drugs, as approximately 25% of QT studies over the past 5 years fall under Q&A 6.1. What about when we do have HERG block and/or QT prolongation? I showed this slide earlier and how drugs that cause TDP are associated with HERG block, action potential prolongation, and QT prolongation. However, it's more complicated. If you look at the bottom, when you have HERG block, there are unopposed sodium and calcium currents that can initiate extra beats, which can cause TDP. This image shows what was on the last slide and how HERG block alone can cause QT prolongation that leads to TDP. However, with certain types of multi-channel block, specifically with HERG block plus either late sodium block, L-type calcium block, or a combination, can cause QT prolongation that does not always lead to TDP because there is a balance of inward and outward ion channel effects. In addition, there are non-ion channel mediated types of QT prolongation, such as from autonomic effects, that do not always lead to TDP. It is important to evaluate these with a non-clinical-clinical integrated risk assessment and to leverage proarrhythmia models. With the stage 1 Q&As, when there is HERG block and/or QT prolongation, follow-up studies can be performed to assess TDP risk. We describe this in the integrated risk assessment Q&As, which reference other Q&As, and the non-clinical follow-up studies can be used to contribute to the design of clinical investigations and interpretation of their results. In stage 1, this is subject to case-by-case evaluation. This leads to the question of whether QT prolongation is still an issue in drug development. Medicinal chemists can engineer out the HERG activity of many new compounds. However, we looked at the past 5 years of clinical QT study reports submitted to FDA, the modern era of QT evaluation, and found that approximately 20% of the study reports found that the drug did prolong QT. It's important to note that this only captures drugs where sponsors have submitted a complete clinical QT study report. So if drugs are dropped in preclinical development or early clinical development where a QT signal is seen, but they don't submit a full report, that's not in this data set. And what types of drugs are they? Probably not surprisingly, about a third of them are oncology drugs, In addition, about a third of them are from drugs that target the central nervous system, and the remaining third is from a combination of different therapeutic areas. Noting that QT prolongation still is an issue in drug development, what does E14 currently state about QT prolonging drugs? The ECG monitoring section indicates that the TQT is not to assess TDP risk, but to determine what further data are warranted to assess risk, and that the intensity of ECG monitoring depends on multiple factors, including characteristics of the drug. It specifically calls out safety pharmacology and pharmacodynamics, which are highly relevant to our new Q&As. In the regulatory implications and labeling section, it indicates that the TDP risk might be influenced by other pharmacologic effects, such as late sodium block or calcium block, and that drugs might prolong QT up to a plateau value above which there is no dose-dependent increase. This can occur with drugs that prolong QT by mechanisms other than direct ion channel block. Overall, this provides a framework for how we could develop more specific Stage 2 Q&As for QT prolonging drugs and how to use safety pharmacology and pharmacodynamic data to assess TDP risk to impact clinical development, regulatory decision-making, and labeling. We propose that in Stage 2, we will describe details on how additional new data can impact specific decision-making. Guidance on follow-up studies and assessments would be provided on how you may use multiple ion channel assessments combined with proarrhythmia models to predict risk, along with assays for mechanisms of QT prolongation beyond direct HERG block. In addition, ECG biomarkers can be used to assess the concordance of in vitro ion channel effects and predictions and clinical ECG effects. What do I mean by that? The ECG and QT interval can be divided into subintervals. We already measure the QRS interval, which is a measure of depolarization, in addition to the PR interval. And repolarization can be divided into the components of early repolarization, J to T peak, and late repolarization, T peak to T end. Analysis of these subintervals can provide insights into the effects of a drug on different specific ion channel currents. It would be important to perform exposure response for QTC and, when appropriate, some of the additional ECG intervals or biomarkers to, as a combined non-clinical clinical integrated risk assessment, indicate how they would impact late phase clinical trial design and regulatory decisions at the time of a marketing application and labeling. We also propose in Stage 2 to define low-risk drugs that do not need detailed QT-focused clinical evaluation and what type of non-clinical evaluation they need to support that. Already, monoclonal antibodies do not require detailed clinical QT evaluation due to the low likelihood of ion channel interactions. Can we expand this to other areas, such as other therapeutic biotechnology products, intermediate-sized proteins, oligonucleotides, and drugs with low systemic bioavailability, and potentially other areas? Each may require different considerations. In summary, our activities to date have focused in Stage 1 on the Q&As that we've developed for S7B on the integrated risk assessment, best practice considerations for in vitro and in vivo assays, and principles of proarrhythmia models. In addition, we have developed the E14 Q&As on how to use the non-clinical data to decrease the need for thorough QT studies and improve regulatory decision-making and labeling when a TQT study or equivalent cannot be performed. Subsequent presentations in this webinar are on the recently released Stage 1 Q&As. We propose that in Stage 2, we would focus on how to use proarrhythmia models and ECG biomarker data to inform decision-making and labeling for QT prolonging drugs, and how to define low-risk drugs that might not require detailed clinical QT assessment. After completing Stage 1 Q&As, the working group will proceed to Stage 2 or make recommendations for what additional data are required in certain areas. After my talk, we will hear about the revised E14 Q&As and presentation of examples to highlight the impact of non-clinical data on clinical development and interpretation. This will be followed by the S7B Integrated Risk Assessment Q&As and considerations for an integrated non-clinical-clinical risk assessment. Finally, we will have a live discussion of questions received from the Q&A pod. This will involve all speakers and some additional members of the ICH E14 S7B Working Group. On day 2, tomorrow, we'll hear a recap and an introduction to day 2, best practice considerations for in vitro studies, best practice considerations for in vivo studies, principles of proarrhythmia models, and again, a live discussion of the questions received from the Q&A pod. The anticipated timeline going forward for the ICH process is that until the end of November, the draft Q&As will be open for public comment in the US and around the same time or just before in other regions around the world. The working group will then review the public comments, potentially update Q&As, and finalize the Q&As. The working group will then finalize technical training material and publish it on the ICH website. And finally, the working group will provide a new timeline and/or recommendation for proceeding with Stage 2 Q&As. I would like to thank all members of the ICH E14 S7B Working Group for coming together to develop these Q&As over the past 2 years and for the time that's gone in to developing the content of this webinar. We want your feedback. Submit your questions during the webinar via the Q&A pod today and tomorrow, and they will be discussed at the end of the presentations each day. In addition, provide official comments during public consultation periods around the world. And now on to Dr. Christine Garnett from FDA, who will discuss the revised E14 Q&As and presentation of examples to highlight the impact of non-clinical data on clinical development and interpretation. Thank you very much.
Good morning, good afternoon, and good evening, and welcome to our webinar on the new approaches for an integrated non-clinical and clinical QT risk assessment. Over the next half hour, I will be presenting the working group changes to the E14 guideline. And in my presentation, I will be using hypothetical case examples to emphasize the changes that were made to the guidelines. So as I go through my presentation, if you have any questions, please submit those questions through the chat box, and I'll answer your questions later today during the Q&A session. So since the implementation of ICH E14 guideline in 2005, the working group has made substantial changes to the guideline through the questions and answers. On the slide, you see a summary of the changes made to the guideline as well as the time frame of those changes. So now that the working group has made additional changes in the use of the non-clinical data to inform clinical decision making, it's important to note that we have not added any new Q&As, but instead decided to modify 2 existing Q&As. And those 2 Q&As are Q&A 6.1, which is the Q&A which describes the use of alternative clinical studies for drugs that cannot undergo a conventional thorough QT study. And the second Q&A that was modified is Q&A 5.1, and this is the Q&A which describes how to use exposure response analysis in early phase 1 studies as a substitute for a thorough QT study. So during my presentation, I am going to be referring to each of these Q&As as Q&A 5.1 and Q&A 6.1. So this slide shows the study reports submitted to the FDA between the years 2016 and 2020. And you can see that most of the QT study reports are coming under 5.1 and 6.1, with only 44% of these studies being a traditional thorough QT study. So the working group is expecting that the revisions to the Q&A 5.1 and 6.1 by using the non-clinical data will increase the number of studies, phase 1 studies, that can be used as a substitute for a thorough QT study. We are also expecting that the non-clinical data will increase the strength of evidence for a low QT risk in alternative study designs under Q&A 6.1. And ultimately, our goal is to decrease the number of thorough QT studies needed in clinical development programs. Before I get into the specific changes to the Q&As, the working group thought it was important that we go over some common terminology used not only in my presentation, but also in other presentations that you'll hear later today and tomorrow. So when I use the term therapeutic dose, this is the dose that's evaluated in phase 3 trial or recommended in the product labeling. And so for our drug X, for this example, let's just call the therapeutic dose 10 milligrams. The clinical exposure for the therapeutic dose is the mean steady-state Cmax associated with the maximum therapeutic dose. And so for drug X, let's just call the mean steady-state Cmax 100 nanograms per ml. So the high clinical exposure scenario, and I'm going to use that term quite a bit in my presentation, this describes the increase in Cmax when the maximum therapeutic dose is given with intrinsic or extrinsic factors. So for drug X, if the exposure increases threefold with the drug interaction, the target for the high clinical exposure scenario is a Cmax of 300 nanograms per ml. A supertherapeutic dose is a dose that provides exposures that cover the high clinical exposure scenario. And so we're looking for a dose that covers that Cmax of 300 nanograms per ml. And for drug X that has linear kinetics, that's about 30 milligrams. And you'll also hear me talk about the dose of the exposures needed to waive the positive control in Q&A 5.1. And we typically take that as twofold the high clinical exposure scenario. So for drug X, if the high clinical exposure scenario is 300, the targeted exposure to waive the positive control is 600 nanograms per ml, which would translate into about a 60 milligram dose for drug X that has linear kinetics. Next, what I want to do is go over some key points in Q&As 5.1 and 6.1 before going into the specific changes that we made to these two Q&As. So Q&A 5.1 is the Q&A that describes how to use exposure response modeling in early phase studies to use as a substitute for a thorough QT study. And so what we're looking for in these studies to be used as a substitute are three key elements. The first element is that the clinical data that characterize the QT response should be at doses that reflect the high clinical exposure scenario. So for drug X that we just went over, the early phase 1 study should at least have a dose that covers that 30 milligrams or that 300 nanogram per ml Cmax. We're also looking that the study incorporates high quality ECGs. We have described what we would like to see as high-quality ECGs in Q&A 1 of the E14 guidelines, but I like to think of it as they should be the same type of ECGs that you typically would use in a thorough QT study. And finally, if a company wants to waive the positive control in that phase 1 study, then we want to see that extra margin to waive that positive control. And so we're looking that the study includes doses that give exposures that are twofold the high clinical exposure scenario. So going back to that drug X that we just talked about, for the phase 1 study to waive the positive control in that study, we'd want to see that the QT was evaluated at doses as high as 60 milligrams, even though the therapeutic dose is at 10 milligrams, and that would be a valid way of waiving the positive control. So for Q&A 6.1, this is the Q&A that describes the alternative clinical study for drugs that cannot undergo a conventional thorough QT study. And a lot of times these are drugs that cannot be given to healthy volunteers, and so the QT effects are evaluated directly in patients. And because we're using patients in that study, a lot of times we may not have a placebo, we definitely will not have a positive control, and a lot of times the QT effects are not evaluated at exposures above the clinical exposures. And due to these limitations, the alternative study does not have the same sensitivity as a thorough QT study. Therefore, in 6.1, there is a description of how to make decisions on the QT risk in alternative study designs. And what is said in 6.1 is that there is reluctance to draw conclusions of a lack of effect in the absence of a positive control. However, if the upper bound of the two-sided 90% confidence interval around the estimated maximum effect on QTC is less than 10 milliseconds, then the treatment is unlikely to have an actual mean effect as large as 20. I know that's a lot of definition, and I like to think of it very simplistically as if the upper bound is less than 10 in this alternative study, then the drug did not cause large mean increases in the QT interval at the therapeutic dose. So now what I want to do is move into the changes that the working group made to Q&A 5.1 and 6.1. And I'm going to start with the changes to 5.1. And so what we've decided in this Q&A is we would like to see that a double negative non-clinical assessment can be used to support a lower clinical exposure needed to waive the positive control. So I thought the first thing we need to do is go over what a double negative non-clinical assessment means. And we have defined that as when the in vitro HERG assay and the in vivo QT study are conducted using best practices and both are negative. Now in my presentation, I am not going to be discussing best practices. This is the topic for discussion tomorrow. But I will give you a definition of negative for each one of these studies. So a negative HERG assay is one where the HERG safety margin of a new drug is higher than a certain threshold calculated based on reference drugs with known Torsade risk using the same HERG assay. And Dr. Jiwa Li from the FDA will be describing this more in his presentation that follows mine. Now, a negative in vivo QT study is one where no significant QT prolongation is observed in the animal studies, where the investigational drug's exposure covers the high clinical exposure scenario. And it's important to note that the sensitivity of this study is different for E14 Q&As 5.1 and 6.1. So for 6.1, we are looking that the study has sufficient power to detect a QT prolongation effect of a magnitude similar to a dedicated clinical QT study, and this requirement is not needed for 5.1. Now, this additional assay sensitivity is needed to support 6.1 or the alternative clinical study, because in the alternative clinical study, that study by itself does not have the same sensitivity as a thorough QT study, so we're going to be relying more on the animal data, and so that's why we want to make sure the animal data does have the sufficient sensitivity as to detect small increases in the QT in the animals. So now with the change of adding this double negative non-clinical assessment, there are now two pathways where a sponsor has available to them to waive the positive control. The first pathway is the original pathway of being able to assess the QT effects at that 2 times high clinical exposure scenario. And if you look at the pie chart on the right side of the slide, this is looking at the QT report submitted to the FDA under Q&A 5.1. It's describing the percentage of studies with different exposure margins. And you can see here that 42% of the study reports submitted under 5.1 to the FDA actually was able to meet that 2 times high clinical exposure scenario to waive the positive control. So with the changes that the working group made to this Q&A, the second pathway is to use that double negative non-clinical assessment, and as long as the clinical data in that phase 1 study can cover the high clinical exposure scenario, then together that would serve as a study that could be used as a substitute for a thorough QT study. And so if you go back to the FDA's experience with studies coming under 5.1, that will allow this additional 30% of the studies to be used as a substitute. So what I want to do now is go over some hypothetical case examples just to illustrate the Q&A 5.1 and the changes that were made. So in the first case, again, this is a substitute for a thorough QT study. The sponsors conducted a single ascending dose study with high quality ECGs. And the highest dose in this study covers twofold the exposure expected when the drug is given with a concomitant strong CYP inhibitor. And so that would be considered the high clinical exposure scenario. And there was no evidence of a concentration-response relationship. So my question to you is, can this SAD serve as a substitute for a thorough QT study under Q&A 5.1? And I will give you a few seconds to think about this before giving you the answer. Okay, I'm going to go ahead and show you the answer. And the answer is yes. And this yes because the study contained those 3 critical elements that we're looking for. We had the QT effects for assessed exposures that cover that high clinical exposure scenario. The that study also included doses that cover that 2 times the high clinical exposure scenario, so they were able to waive the positive control. And the study included high quality ECG. So this is a perfect example of what we're looking for a study that could serve as a substitute for a thorough QT study using that one pathway to waive the positive control. So let's look at a variation of this request. Let's say the sponsor conducted an SAD study with high quality ECGs. However, in this case, the highest dose only covers the exposure covered when the drug is given with concomitant strong CYP inhibitor. So the exposures cover the high clinical exposure scenario. However, the exposures are not high enough to waive the positive control. So now the sponsor can use this new pathway to waive the positive control by including their non-clinical assessments. So if further non-clinical assessments, the HERG safety margin was greater than 100 using best practices for both parent compound and their metabolite, but they didn't submit their reference drugs. And for their non-clinical in vivo study, they didn't see any QT signal in dogs at exposures that were greater than the high clinical exposure scenario, but the study was not powered to detect prolongation similar to a thorough QT study. So my question to you is, can this integrated assessment serve as a substitute for a thorough QT study under the revisions to Q&A 5.1? And again, I'll give you a few seconds to think about the answer before I show you. Okay, I'm going to go ahead and show the results. So the answer to this question is no, and let me go over the reasons why. So the non-clinical study, first of all, we're looking for that double negative assessment. Now the HERG margin was large, it was greater than 100, but the sponsor did not show that that large margin was actually larger than the safety margins for reference drugs with the known Torsade risk. Using the same assay. This requirement or this suggestion is going to be described more in S7B Q&A 1.2. And this is illustrated on the figure to the right of the screen. What we're looking for is that HERG margin for the new drug is going to be larger than the HERG margins for these reference drugs. And Dr. Jiwa Lee from the FDA is going to be talking to you more about this in his presentation that follows mine. Now for the in vivo study had the exposures exceeding the high clinical exposure scenario, and that's what's key to support 5.1, and we're not necessarily looking that the study is powered to detect QT prolongation similar to the thorough QT study. And Dr. Sonoda is going to be talking about this in more details tomorrow when he discusses S7B Q&A 3.4. So now that I've gone over the changes for 5.1, I'm going to now switch over to talk about the changes that we've made to Q&A 6.1. Now for 6.1, what we're looking for is that this double negative non-clinical assessment can now be used to support an alternative clinical study to show low QT prolongation risk. So when I went back and looked at the FDA database of QT reports submitted under Q&A 6.1, what I find, and which is illustrated in the pie chart, is that 80% of those studies are actually conducted for drugs being developed for oncology indications. So these alternative study designs are mainly conducted in cancer patients where there's no placebo, there's no positive control, and there's really no super therapeutic doses. A lot of times these studies are conducted using the therapeutic dose. So because of these limitations in these alternative study designs, it's really difficult to demonstrate lack of a QT effect. So now what we've done to the 6.1 is that the working group has used the non-clinical data to support a drug having a low likelihood of a proarrhythmic risk due to QT prolongation. And the revisions have stipulated that in order to show low risk for QT prolongation, that the data have to meet 3 criteria. The first is that the non-clinical data meet that double negative assessment where the HERG margin for the parent and metabolites are conducted under best practices and higher than the threshold. And this is exactly the same as under 5.1. But what is difference is between 6.1 and 5.1 is this in vivo study, because the clinical data are not as sensitive in this alternative study as a thorough QT study, that we're looking for that sensitivity in that in vivo study. So the animal study should be powered to detect QT prolongation out of a similar magnitude to a dedicated clinical study. Again, this is described in S7B Q&A 3.4, and it'll be discussed more tomorrow. We're also looking, just like with 5.1, that the exposures in the animals are great, or at least cover that high clinical exposure scenario. The second revision that we made to 6.1 is in the second criteria to be met is in that alternative clinical study. We are expecting that high quality ECG data are included, and the analysis do not suggest QT prolongation. And we've now changed the definition of QT prolongation in this Q&A. We are defining it as a mean baseline corrected QT less than 10 as computed either by the exposure response or intersection union test. This will depend on whatever the sponsor think is appropriate for their study design. And the strength of clinical ECG data will depend on the upper bound of the two-sided 90% confidence interval around that mean estimate. And so what we're saying here is that we will look at the magnitude of the estimate, but the decision making won't focus so much on that upper bound because we're going to be informing that study with that non-clinical data. And finally, we'll also be looking within that study that there are no notable imbalances between the treatment arms and the proportion of subjects that have QT outliers. And the final criteria to be able to show that the drug has low QT risk is that when the clinical database is complete at the time of marketing application, that there's no increased rate of adverse events that would be a signal for proarrhythmic events. So we'd also be looking for that. So let's go over, again, some hypothetical cases just to reinforce the changes to 6.1. So here, the sponsor has conducted an uncontrolled dose escalation study with an expansion cohort for an oncology drug in 75 patients. And the study included high-quality ECGs. The highest dose studied was the labeled dose, and there was no evidence of QT prolongation using exposure response analysis, where the mean QT effect at the Cmax was 4 with the upper confidence interval of 8, and there were no outliers detected. Now, the sponsor in this case example did not conduct safety pharmacology studies according to best practice, and therefore the low QT risk assessment would have to be based solely on the clinical data. And so can the clinical QT assessment support low risk for QT prolongation under 6.1? Again, I'll give you a few seconds to think about this before I give the answer. Okay, I'm going to go ahead and show the answer. And the answer is no, and let me tell you why. First, none of the non-clinical studies were not conducted to best practices, therefore we're not going to be using the non-clinical data to inform the clinical decision making. And when relying only on the clinical data, there is reluctance to draw conclusions of a lack of effect in the absence of a positive control, a large exposure margin. Therefore, what you could conclude from this study is that the drug did not cause large mean increases in the QT interval at the therapeutic dose, but you would not be able to exclude small effects based on the clinical data by itself. So let's go over a variation of this case for another oncology drug 2. And so here, the sponsor conducted an uncontrolled dose escalation study with high-quality ECGs, and this time it's in 30 patients. Again, the highest dose studied was the labeled dose, and there was no evidence of QT prolongation using the new definition, which the mean estimate was 4, but because there was low number of subjects, the confidence interval popped up to 12. So exceeded that 10 millisecond regulatory threshold. And there were no QT outliers detected in that study. Now, the sponsor in this case did conduct the safety pharmacology studies according to best practice. The results showed double negative assessments, and they were able to conduct their in vivo study with sufficient power. So now I ask you, can this integrated risk assessment support low risk for QT prolongation under the revisions to Q&A 6.1? Again, I will give you just a few seconds to think about your answer. All right, I'm going to go ahead and show you the answer. And the answer is yes. First of all, the clinical QT study had high quality data and did not show QT prolongation according to our new definition of that, which meant that the point estimate was less than 10 and there was no imbalance of the outliers. And the strength of the evidence of that estimate was pretty good, and we're going to look at that at the confidence interval. It just slightly increased over that 10 millisecond threshold. And finally, which we really wanted to see, is that there was a double negative non-clinical assessment. So the working group is very interested in your thoughts about how we changed 6.1. And so if you're planning on providing comments to the Q&As during the public comment period, we are specifically calling out this new definition, and we'd like to seek your opinion on whether you think the way we are defining the lack of clinically relevant QT prolongation in the context of the alternative study designs, when we know there is a double negative non-clinical assessment and there's no evidence of proarrhythmia in the clinical database. So now I'd just like to conclude my presentation by summarizing the changes that were made to the Q&As. So for 5.1, what the working group has done is now suggesting we could use a double negative non-clinical assessment to support a lower clinical exposure needed to waive the positive control. Again, for that double negative non-clinical assessment, we're looking that the HERG safety margin is greater than the reference drugs used that were evaluated using the same assay, and also that the in vivo QT study evaluated doses covering the high clinical exposure scenario. And for 6.1, again, the double negative non-clinical assessment can now be used to support an alternative clinical study to show low QT prolongation risk. The double negative non-clinical assessment, the HERG margin should be the same as 5.1. However, the in vivo QT study, we want to make sure that it covers that high clinical exposure scenario, but the study also needs to be appropriately powered to detect QT prolongation similar to a thorough QT study. And so with that, I'm going to conclude my presentation. I hope that I have answered any questions that you have about the changes to the guidelines, and that I provide you sufficient information that you're able to implement these changes within your organization and to use the non-clinical data in your clinical decision making. Next on today's agenda, you're going to hear more about that double negative non-clinical assessment and how to do that HERG margin from Dr. Zihua Li from the FDA, and he's going to be presenting this S7B integrated risk assessment Q&As. I thank you again, and I look forward to talking to you during the Q&A session.
Thanks, Christine, for the introduction. Hello, everyone. Welcome to this webinar. Today, I will give a presentation about the new Q&As for S7B integrated risk assessment. ICH E14 was first established in 2005 after multiple drugs were removed from the market due to Torsade or TDP. S7B is about non-clinical cardiac safety pharmacology that includes HERG potassium channel block, non-clinical in vivo QT study, and follow-up studies as needed. S7B is successful in keeping study participants safe in early clinical trials. However, there is room for improvement. In S7B, there is limited guidance on some experimental details, which has led to variability in assay performance. And as clinical development continues, non-clinical studies are largely ignored, and clinical safety assessment relies on human QT, which is an imperfect biomarker. So ICH is developing Q&As to address these issues. As David indicated, the Q&As will come in two stages. As of now, the draft Stage 1 Q&A has been released. Shown here in the center is a flowchart from the current S7B guideline. The new draft Stage 1 Q&A covers different parts of this flowchart, including integrated risk assessment considerations, best practice considerations for ion channel assays and in vivo QTSAs, and best practice considerations for mild-sized assays, and principles for Proarrhythmic models. My presentation today will be focused on integrated risk assessment considerations. In the Q&A, we have two questions dedicated to that topic. Question 1.1, what is the general strategy for use of non-clinical information as part of an integrated risk assessment for delayed ventricular repolarization and Torsade palm that can inform the design of clinical investigations and interpretation of their results? Question 1.2, what is the recommended method to compute the HERG safety margin? So let's take a look at the overview of the non-clinical strategy. The Q&A describes two scenarios to use non-clinical data to inform clinical decision-making. One is the so-called double negative scenario when the in vitro HERG assay and the in vivo QTSA are negative. Another one is the non-double negative scenario when the in vitro HERG assay and or in vivo QTSA are not negative. Of course, before we describe these two scenarios, we need to first define what is a negative in vitro HERG assay and what is a negative in vivo QTSA. Principles to define these negative assays are provided in the Q&A. In the next two slides, I will summarize those principles. One point to make is that current ICH S7B guideline does not specify how to define a negative co-assay, so this is something new in the Q&A. First, principles to define a negative HERG assay. We say a drug is HERG negative when its safety margin, usually calculated as HERG IC50 over Cmax, is higher than the safety margin determined based on reference drugs known to cause TDP. Experimental variability should be incorporated in HERG safety margin calculations. In other words, IC50 variabilities should be translated to a confidence interval around the safety margin. HERG IC50s should be determined following Q&A 2.1 best practice considerations. The same experimental protocol should be applied to the new drug and the reference drugs. Here, Cmax is defined as mean steady-state maximum plasma concentration when the maximum recommended therapeutic dose is given with intrinsic or extrinsic factors. This is also referred to as high clinical exposure scenario by Christine earlier. High clinical exposure will be an estimate early in development that is subsequently refined. Free or unbound fraction of plasma concentration is usually used. Free fraction should be set to 1% if experimentally determined to be less than 1%. This is consistent with FDA guidance document on drug interaction studies. If protein binding cannot be accurately assessed or tissue levels may exceed free plasma concentrations, both free and total Cmax should be used. So these are the principles to define a negative HERG assay. What about principles to define a negative in vivo QT assay? We say a drug is in vivo QT negative when no QT prolongation in animal studies were observed, when the new drug's exposure includes and exceeds anticipated clinical exposure. Both the parent and any major human metabolites need to be considered. The experiments should follow general in vivo best practice considerations, such as species selection, heart rate correction, and reporting format, which will be discussed in detail tomorrow. To support E14 Q&A 5.1 and 6.1 situations, as discussed by Christine earlier, exposures should cover the anticipated high clinical exposure scenario. The adequacy of exposure to any major human-specific metabolites should be determined. And to support E14 Q&A 6.1 situation, the in vivo study should have the power to detect a QTC prolongation of a magnitude similar to a dedicated clinical QT studies. Now we have principles to define both in vitro HERG assay and in vivo QTSA. We can take a look at the double negative scenario. The double negative scenario can be used to support two clinical situations. One, to lower clinical exposure needed to waive the positive control. This is the E14 Q&A 5.1 situation. And two, to support alternative clinical QT study design. This is the E14 Q&A 6.1 situation. For both, the in vivo QT study should cover the anticipated high clinical exposure. And for 6.1, in addition, the in vivo QT assay should be powered to detect QTC prolongation of a magnitude similar to a dedicated clinical QT studies. Now let's take a look at two examples, one for E14 Q&A 5.1 and one for 6.1. First, let's say a sponsor conducted first-in-human study with high-quality ECGs. The highest dose covers the exposure expected when the drug is given with concomitant strong CYP inhibitor. This is one example of the high clinical exposure scenario. Concentration QT analysis rules out 10 millisecond effect at high clinical exposure and no QTC positive control. Question is, can the first-in-human study serve as a substitute for TQT study? Under the current guideline, the answer is no, because there is no QTC positive control, and the exposure in the clinical study is not high enough to waive the positive control. But with the new Q&A, the sponsor can submit non-clinical data. Let's say for the in vitro HERG data, the sponsor first showed that the assays were conducted following the best practice considerations, which will be discussed in detail tomorrow, and also other considerations which I summarized as principles to define negative in vitro HERG assay earlier. The safety threshold was determined using reference drugs with known TDP risk, and the sponsor submitted a justification of choosing this threshold and reference drugs. We can see in this figure that the sponsor chose a few reference drugs, the red drugs known to cause TDP. They applied some HERG protocol to these drugs, and they calculate the safety margin IC50 over Cmax. The error bars are 90% confidence interval of safety margin for each drug. Then they determined a safety threshold, the dashed vertical line, to be having high sensitivity to detect torsadogenic drugs based on the reference drugs. Then, with the same experimental protocol, the new drug safety margin was determined to be above this threshold, as shown by this green drug here. For the in vivo QTSA, the sponsor first showed that the assays were conducted following the best practice considerations, which again will be discussed in detail tomorrow, and no QTC prolongation at the concentrations of parent, compound, and human-specific metabolites that cover high clinical exposure. Now, with all this clinical and non-clinical data, the integrated risk assessment can substitute for a TQT study. This is only possible with the new Q&A. Now let's take a look at the second example for E14 Q&A 6.1. Let's say the sponsor conducted an uncontrolled dose escalation study with expansion cohort for an oncology drug. The highest dose studied was the labeled dose. No evidence of QTC prolongation using concentration response relationship and no important QTC outliers. Cardiac safety database at the market application does not suggest proarrhythmia risk. So the question is, can the clinical QT assessment support low risk for QTC prolongation or TDP? The answer is no under the current guideline, because this is an alternative clinical QT study design without placebo control and without other factors. However, with the new Q&A, the sponsor can submit non-clinical data. Let's say the in vitro HERG data or the same as the previous example with E14 Q&A 5.1 is negative. For the in vivo QTSA, the data are the same as before, plus demonstration that the in vivo QTSA was powered to detect QT prolongation of a magnitude similar to a dedicated human QT study. Now, with all this clinical and non-clinical data, the integrated risk assessment supports the clinical interpretation of low TDP risk. Again, this is only possible with the new Q&A. Now, both of these two examples are about double negative scenarios. What about non-double negative scenario? If a drug is not double negative, then follow-up studies may be performed to further evaluate TDP risk on a case-by-case scenario. Best practice considerations for some follow-up studies are described in S7B Q&As, such as additional ion channels, human-derived cardiomyocytes, and proarrhythmia risk prediction models to quantify TDP risk level. You will hear discussions about these studies in detail tomorrow. Follow-up studies are optional. If a drug is not double negative, the sponsor does not need to perform follow-up studies. However, if they choose to perform follow-up studies, sometimes they can be used to inform clinical decision-making. Let's take a look at one example. Let's say sponsor conducted first-in-human, both single and multiple ascending dose studies with high-quality ECGs. Highest dose evaluated covers the high clinical exposure scenario. The exposure response data rule out 10 millisecond QTC prolongation and suggest no relationship between concentration and QTC, and no relationship between concentration and PR or QRS intervals. There's no positive control for QTC. Because there's no positive control and the exposure is not high enough to waive a positive control, the sponsor conducted non-clinical core assays. For the in vivo QTSA, the results are the same as the E14 Q&A 5.1 example earlier, so it's negative. For the in vitro HERG assay, the drug's safety margin is slightly lower than the safety threshold based on reference drugs. Even though this drug looks like a borderline drug, by definition, this drug is not double negative. So, the sponsor voluntarily conducted follow-up studies. They performed additional ion channel studies, which suggested that the drug blocks both IKL HERG and an inward current. For example, the inward current could be base sodium current and or L-type calcium current. The sponsor also used a qualified in silico model to quantify the block effects and predicted the drug to have a low TDP risk. How to qualify models for TDP risk prediction will be discussed in detail tomorrow. The sponsor also used a cardiomyocyte assay and found no proarrhythmia events with the new drug. Now, with all this clinical and non-clinical data, including follow-up studies, the outcome is that only routine safety ECG monitoring is needed in later development. One point to make is that such an algorithm to use follow-up studies is not written in the Q&A, and it is subject to case-by-case evaluation. Now, all these previous three examples are about Stage 1 Q&A. As David indicated, in Stage 2, S7B and E14 Q&As will be developed on how to use proarrhythmia prediction models or algorithms to influence the design of late-phase trials and to inform labeling for QT prolonging drugs. Even though the Stage 2 Q&A is not released yet, the integrated risk assessment, including the results from follow-up studies and other relevant clinical and non-clinical information, can contribute to the design of subsequent clinical investigations and interpretation of their results. This is a statement first stated in the S7B guideline and reiterated in the draft S7B Q&A 1.1. So follow-up studies can be used on a case-by-case scenario to evaluate the risk for these drugs. In summary, S7B should continue to be followed for obtaining non-clinical data to support first-in-human studies. The new integrated risk assessment Q&As provide additional recommendations when non-clinical data are used later in clinical development. However, applying the best practice early on is encouraged, and that might prevent repeat assays during clinical development. Double negative non-clinical assessments can be used to support E14 Q&As 5.1 and 6.1, and optional follow-up studies can be used to further evaluate QT or TDP risk when non-clinical core assays are not negative. I would like to thank all members from the ICH E14 and S7B Working Group. And that's all for my presentation. Thanks for your time. Next, Jean-Pierre Valentin from FPA will be discussing considerations for an integrated non-clinical and clinical risk assessment. Thank you.
Offering. While at adoption, ICH E14 suggested the QT interval evaluation independent of the ICH S7B result, both documents highlight the need for integration of information in a manner which is informative of the totality of evidence. The current ICH activities are directed towards scenarios where the non-clinical data are informative in clinical study implementation and evaluation. Therefore, the question being asked is how can an integrated non-clinical to clinical risk assessment be informative for clinical development, for regulatory decision-making, and for labeling. This slide reflects the evolution of the QTC Torsade point or TDP landscape in drug development pipeline over the last 20 years. Over the last 2 decades, there has been a shift in portfolio modalities entering drug development. In particular, there has been an increased proportion of non-small molecule candidates entering development, such as biotechnology-derived products. There has been an increase in the proportion of oncology products. In relation to QT-TDP assessment, there has been an increased usage and acceptance of the concentration QTC modeling as a substitute for the clinical thorough-QT study. There has been an increase in the proportion of TQT negative. And lastly, there has been a gradual reduction in the incidence of QT-TDP-related adverse events in the FDA adverse event reporting system. Altogether, these facts are suggestive of a positive impact of the ICH S7B and E14 in relation to QTC-TDP risk assessment in drug development. This slide depicts a prototypical drug discovery and development workflow for a small molecule program. It starts with the identification of a target of potential therapeutic benefit, all the way through identification of a clinical drug candidate, entering and progressing through the clinical development phases. The workflow encompasses the design, synthesis, and testing of thousands up to millions of compounds in relevant assays, and the selection of a drug candidate with suitable property to be administered safely to humans. It is a lengthy, costly process associated with a low success rate. Therefore, any novel approaches that may contribute enhancing the likelihood of success whilst benefiting and protecting patients should be encouraged. And that's what is being attempted by these Q&As. So what is the value proposition and anticipated impact of the new E14 S7B Q&As? It is based on the use of high-quality non-clinical data in order to, first of all, make better informed decisions earlier in drug development by streamlining the drug development process, by reducing or eliminating the burden on clinical studies. Secondly, to inform clinical development and reduce the need for dedicated thorough study by supplementing phase 1 ECG evaluation when exposure margin is insufficient to waive positive control in a concentration response analysis. Reference to E14 question and answer 5.1. And thirdly, to change regulatory decision and labeling by supplementing QT assessment when a specific TQT study cannot be conducted. Reference here to the E14 question and answer 6.1. What do we mean by non-clinical to clinical integrated risk assessment? This cartoon illustrates the concept of an integrated risk assessment. It is an integration of increasingly complex, costly, and lengthy information throughout the pharmaceutical life cycle. It starts very early in the drug discovery process as soon as relevant information arrives relating to cardiac repolarization. The integrated risk assessment is closely related to the benefit-risk concept that is dependent upon a number of elements, such as but not limited to disease indication, patient population, and standard of care. Let's now review how the component of the QT integrated risk assessment has evolved over the years, starting with the original, i.e. post-2005, component of the QT integrated risk assessment, or as it was the case at the time, a non-integrated risk assessment. It is fair to say that the different pieces of the QT integrated risk assessment are aimed at supporting key regulatory decision points, which are fixed in time in relation to drug development. These include the first investigational new drug submission, the late-stage clinical development entry, and the new drug application. The integrated risk assessment starts typically by taking into account data generated during the early discovery phase. These are typically non-regulated, not conducted to a good laboratory practice standard, and are sponsor dependent. Consequently, the degree of knowledge and confidence in an asset varies considerably from sponsor to sponsor. At the time, and from a regulatory point of view, the assay results from the S7B, comprising the core battery in vitro HERG and in vivo QTC assay, were not considered as part of the overall integrated risk assessment. So to recap regarding the original component of a QT non-integrated risk assessment, the regulatory decision points are fixed in time relative to clinical development phases. The original S7B and E14 information were not explicitly linked and not considered as part of a non-clinical to clinical integrated risk assessment, but they were also largely focused on different regulatory decision points. So, learning from the past, the goal of the new question and answer is to inform on delayed ventricular repolarization, QTC prolongation, and TDP risk in early and late clinical development based on the totality of evidence. The integrated risk assessment evolves over the years, in particular following the introduction of the concentration QTC analysis conducted in phase 1. The phase 1 concentration QTC analysis enabled several options, including the conduct of the standard TQT or a TQT substitute, although under strict conditions with narrow requirements, and overall had limited impact on decision making, in particular when a TQT could not be performed. That said, the non-clinical and clinical information are still not explicitly considered as part of an integrated non-clinical to clinical risk assessment. Consequently, only the clinical ECG data is used to inform ECG monitoring in phase 3 and to inform regulatory decision-making and labeling. So to recap regarding the current component of a QT non-integrated risk assessment, The phase 1 concentration QTC analysis does enable for TQT substitute, although under strict condition with narrow requirement. However, the non-clinical and clinical information are still not explicitly considered as part of a non-clinical to clinical integrated risk assessment. How can we operationalize the proposed Q&As to form a fully integrated risk assessment? The Q&A concept paper states that one should consider the impact of his non-clinical recommendation on clinical situation where current E14 methodology is problematic. For example, clinical QT assessment that are confounded by issues such as heart rate changes, inability to test a supratherapeutic concentration, and absence of a placebo group. Furthermore, the use of an integrated non-clinical to clinical risk assessment to impact clinical ECG monitoring should be considered under question 5.1, question and answer 5.1, i.e. use of clinical concentration QTC modeling when maximum therapeutic exposure has not been fully covered clinically. And question and answer 6.1, under scenarios where a placebo control comparison is not possible, such as oncology products. There may be potentially other circumstances yet to be defined, for example, low systemic exposure drugs or highly selective drugs. The schematic presented on this slide depicts the proposed component of a QT-TDP fully integrated risk assessment under the new E14 question and answer 5.1 with supporting non-clinical data. Under those circumstances, the use of non-clinical data as defined in the new Q&As, together with the use of phase 1 clinical concentration QTC modeling, would enable for a TQT substitute under expanded condition taking into account non-clinical data. Such dataset would enable to inform ECG monitoring in phase 3 and to inform regulatory decision making and labeling. The schematic presented on this slide depicts the proposed component of a QT-TDP fully integrated risk assessment under the new E14 question and answer 6.1, i.e. when a TQT or a TQT substitute is not possible. Scenario includes situations where a placebo control comparison is not possible, such as oncology drugs. Under an ICH S9 or a standard S7B paradigm, the non-clinical data had no influence on the clinical monitoring. Furthermore, the clinical monitoring that is achievable has limited impact on regulatory decision-making and ultimately on labeling, i.e. only enabling to exclude large QTC effect. Under the new Q&As, the integration of high-quality non-clinical data and phase 1 concentration QTC analysis will enable to be informative in terms of subsequent clinical QTC monitoring, regulatory decision, and ultimately labeling, offering opportunities to be for a less unequivocal, clearer labeling such as no QTC or TDP risk. To summarize the proposed component of a QT fully integrated risk assessment, the current S7B remain adequate to support first-in-human regulatory decision point, i.e. the initial IND submission. The S7B Q&As offers an opportunity to further reduce clinical QT-QTC monitoring under certain circumstances based on the totality of evidence. Under question and answer 5.1, i.e. to use double negative non-clinical data to supplement phase 1 ECG evaluation when exposure margin is insufficient in order to reduce TQT clinical studies. And secondly, under the Q&A 6.1, to supplement QT assessment when a specific study cannot be conducted in order to change regulatory decision-making and labeling. What's the future state for a QT-TDP fully integrated risk assessment might look like? 3 elements are worth considering as part of the next stage. Firstly, to define the drug that may not require detailed clinical QT evaluation, such as low bioavailability drugs or highly selective biotechnology-derived product. Secondly, to optimize and expand integrated risk assessment to other scenarios, in particular when all non-clinical and early clinical QT-TDP related data are negative, by reducing even further the late-stage ECG monitoring whilst informing regulatory decision and labeling. Thirdly, to refine the use and impact of proarrhythmia model and ECG biomarker data to inform decision-making and labeling for QT prolonging drugs. To recap on the future state of the QT-TDP integrated risk assessment, we can ask several questions. First, can low-risk drugs that might not require detailed clinical QT assessment be defined? Second, can non-clinical to clinical QT-TDP integrated risk assessment be further optimized for other scenarios? And third, can we refine the use and therefore ultimately the impact of proarrhythmia model and ECG TDP biomarker to inform decision-making and labeling for QT prolonging drugs? In summary of this presentation, several points can be captured. First, the regulatory decision points are fixed in time relative to clinical development phases. Originally, the S7B and E14 information were not explicitly linked and not considered as part of a non-clinical to clinical integrated risk assessment. But they were also largely focused on different regulatory decision points. The 2015 question and answers focusing on concentration QTSA analysis, and now the 2020 question and answers do offer opportunities to make data collection and integration more efficient, more focused, and more impactful. Alternative development pathway exists for monoclonal antibodies and oncology agent. Question and answer 6.1 provide an option for oncology product to reduce clinical burden and impact the label. One could ask the question whether the monoclonal antibody approach could be expanded to other modalities. This is the end of my presentation, and I will now hand over to the discussion of questions received from the Q&A pod. Thank you very much.
Thank you for those wonderful presentations. We're going to take a 1 or 2 minute break to review the questions you submitted. If you have further questions, please submit them in the Q&A pod in the lower right-hand corner. And we'll be back with the Q&A panel in just about 2 minutes.
FDA. I am the rapporteur of the ICH E14 S7B Working Group, and I'm joined here during the live question and answer period by all of the presenters, and that includes Christine Garnett from FDA, Ziwa Lee from FDA, Jean-Pierre Valentine from FPIA, along with some additional panel members. That includes Colette Stranova from Health Canada. That includes Derek Leishman from Pharma, and that includes Flora Musumaba from EC Europe, EMA, and Charles Benson from FPIA. We have received many questions that have come in during the webinar. Thank you very much to everyone for actively participating and submitting questions. We have had to combine some of the questions together, and there are some questions that focused more on topics that will be discussed in more detail tomorrow. So some of them have been deferred and will be addressed in presentations and then could be asked again tomorrow. With that, I'm going to start going through the different questions, and we'll be directing them to the different presenters or additional panelists to respond. The first question we got referred to, and this will be directed to Christine Garnett from FDA. The question highlighted that in E14 Q&A 5.1, to conclude that a expanded ECG safety evaluation during later stages of drug development is not needed, or in 6.1, E14 Q&A 6.1, to support a drug as having a low likelihood of proarrhythmic effects due to delayed repolarization. We'd like to clarify whether in 6.1 this is interpreted by regulators as a substitute for a negative thorough QT study. Christine, would you like to elaborate on this question?
And excuse me, David. We just discovered that during the internet session there, Christine is no longer in the room, so you may want to go on to another one of the presenter questions while we try to get her reconnected.
Okay. Charles Benson, I see you are in the room, so I'm going to direct a question to you. The question that came in was, if HERG is considered positive, should a sponsor conduct a standalone TQT study for QT characterization? Charles, could you respond to that question? Charles, we're not hearing you. Okay. I'll go ahead and give a response to that question. So when HERG is positive, we generally recommend, a standalone thorough QT study or an alternative approach using phase 1 first-in-human concentration response modeling can be used to characterize the potential for QT prolongation in accordance with E14. So you do not need to do a standalone TQT study. You can also use concentration response modeling in phase 1 as outlined in E14 Q&A 5.1. All right, I'm going to go on to a question, and Jeff, if you could pull up slide 93 in the supplemental deck. The question is, I'm somewhat concerned the presentation further reinforces a HERG-centric model of QT prolongation, and this is misleading as there are many other ways to prolong the QT interval. This was something that was commented on in my slides. and it is commented on in the ICH S7B integrated risk assessment Q&As as well. And if we could go one slide further in that deck to 93, we recognize that there are other methods to prolong the QT interval that can be from autonomic effects, other indirect effects. In the S7B integrated risk assessment Q&A, we discuss how these can be investigated in follow-up studies, and so we do not view the Q&As as being completely HERG-centric, although the core S7B studies have not changed in terms of assessing HERG block and the potential for QT prolongation. And that slide is coming up, and it does show how you can have different types of QT prolongation, HERG block alone, HERG block with other ion channel block that does not necessarily lead to Torsade point, and other types of non-ion channel mediated QT prolongation. Okay, I'm going to go on to another question that Charles, can you comment if you are able to respond?
I hope so. Can you hear me now?
I see you. Now I can hear you. Great.
The issue earlier.
Okay, so no problem. So I'm going to, okay, the new question we have is, when using early clinical data, not all intrinsic and extrinsic factors, metabolites, or the final clinical dose also may not be known. Would it be possible to give more guidance on what would be considered acceptable as an exposure range or window to be covered?
So this is one that's been coming up since we published the Q&A back in 2015 on using first-in-human studies. And it's certainly true that all metabolites or intrinsic, extrinsic factors, and even the final clinical dose might not be entirely understood at the time that the first-in-human study is conducted. So therefore, the fulfillment of the E14 exposure requirements in both 5.1 and 6.1 are caveated. That is, the exposure required to meet the criteria are estimated until the final intrinsic, extrinsic factors in clinical doses are known. So then we argue that at that time, the E14 has been followed when the final biopharmaceutics package metabolites and clinical doses are known. And so this really provides additional motivation for sponsors to fully explore dose ranging in phase 1. So the bottom line is push the dose in phase 1, do your best estimates of extrinsic, intrinsic factors, metabolites, and clinical dose, and then make your arguments when those are finally done.
Okay, thank you, Charles. While we have you here responding, I'm going to ask another question to you. And then I understand Christine Garnett is back connected, and we'll go back to some questions for Christine. But for Charles, for the double negative non-clinical scenario, how do you handle molecules where in vitro HERG assays are not performed due to the molecular entity? And referencing, the question references, for example, oligonucleotides, peptides, monoclonal antibodies, etc. And is the default that they are considered HERG negative? And so I think we have want to there's some nuances that question to divide out. Charles, could you
well, I'll do my best. So this was taken on in a Q&A a couple rounds ago on large molecule exploration of QT characterization. The problem with that particular Q&A is that we did not state what the cutoff is for large molecules. Currently, the interpretation is that large molecules are antibodies, and there's still ongoing discussion on those other oligonucleotides or polypeptides that as to whether they qualify as large molecules or small or intermediate. Certainly, we think that the blockade of the HERG channel is quite important, and if those molecules are so large that they're unable to block the HERG channel, then it is unlikely that they will cause significant issues from a Torsade's standpoint. But this is something that is still ongoing in discussion, and as Dr. Strauss referred to in his first presentation, it is something that we plan to take on in phase 2 of the Q&As that we're putting together.
Thank you, Charles. Christine Garnett, I'm going to direct a question to you. Can you let us know if we can hear you?
Sure.
Can you
say
hear me? Something? I can. Terrific. Okay, well, I'm going to rephrase that first question that we brought up, and this focused on E14 Q&A 6.1. And can you elaborate on what is required to support a drug as having a low likelihood of proarrhythmic effects due to delayed repolarization under Q&A 6.1?
Sure. So this is what's new in the 6.1 Q&A. So what we've done is we're asking companies if they cannot do a study directly in healthy volunteers, then they have to have, because they have a molecule that goes through the 6.1 pathway, what we're looking for is the double negative non-clinical assessment. And in the alternative clinical study, we would still like it to be as, include as many of the traditional thorough QT study designs as possible, understanding there'll be limitations, either drug-specific or patient-specific limitations of that design. Include that, but we're also looking that the database for the marketing submission of the application, that the cardiac database across all trials is not giving an imbalance of adverse events that would be considered proarrhythmic. So we're looking for those three components. Now, of course, one of the questions you might be asking is, well, what do we do in phase 3? Does this mean that all phase 3 studies have to have ECG recordings, and that's not necessarily true. If the sponsor has a double negative in their non-clinical and they do this alternative QT assessment and they're not detecting any QT prolongation, then I think together that would probably mean that they don't have to do additional ECG monitoring in their phase 3 program, just like you would do if you had a negative thorough QT study or you have a negative result in an alternative study design under 5.1. So I think together that could also, the non-clinical double negative plus the alternative clinical study design that does not show QT prolongation, together that would probably bring a package that would cause a company just to have to collect routine monitoring in their phase 3, and you wouldn't have to do an expanded ECG monitoring.
Thank you, Christine. The next question I'm going to ask Dr. Flora Mutsamba from the EMA to respond. If by any chance there's a problem with audio, then Christine Garnett can jump in to respond. The question is, can you please explain why the new approach to use non-clinical data to inform decision-making is only available in cases in which a thorough QT study or substitute cannot be performed? Or why would this approach be considered insufficient if applied to other drugs meeting the double negative standard? Flora, are you able to comment on that? Okay, we're not hearing from Flora. Christine Garnett, would you be able to respond to that question?
Sure. So With the two Q&As, we have the two scenarios where you have a double negative non-clinical assessment, which was going to support the clinical QT assessment. In 5.1, it's really supporting that exposure margin to make sure that so you can waive a positive control. In 6.1, because the clinical alternative study design does not have the same level of sensitivity because it might not have a placebo control, doesn't have large exposure margins, that we're going to rely more on the data from the non-clinical studies, particularly that in vivo animal study, to give us reassurance that the drug does not have a proarrhythmic signal. And so, so those are the two pathways to support the non-clinical data. Now, the question is, if you have a new drug and a sponsor has elected to do that include that additional sensitivity in their in vivo animal study, so they're going to have they have a double negative non-clinical assessment plus the animal study is powered to detect small QT effects, and they want to go down 6.1 pathway, they could, but I would recommend they speak to the regulatory agencies to make sure everyone's in agreement on, especially on the exposure margin. So there's nothing preventing a new compound to go down the 6.1 pathway. I would just recommend, if that's a strategy that a company would like to take, that they do speak to the regulatory agencies about that.
Thank you, Christine. The next question is going to be for Ziwa Lee. It seems that the revised Q&A only takes into consideration HERG and in vivo data. What about mixed ion channel blockers where the proarrhythmia in silico model, that the proarrhythmia model ranks as low risk? These compounds may not be HERG negative, and in this case, how do we handle those? And this question did come in before Ziwa Lee's presentation, where he discussed this. But Ziwa, can you add a little bit to this, the response to this question?
Sure. Actually, the stage 1 Q&A considers non-clinical studies that include not only HERG and the in vivo, but also follow-up studies. For example, in the third example in my presentation, we discussed a scenario where a drug is not double negative. The HERG is positive, even though it's borderline, it's still positive. But then, in that hypothetical example, the sponsor can use follow-up studies that include qualified models to predict risk, additional ion channel studies, and other myocyte assays to prove that the drug is actually of low risk. So that's one example where you can use follow-up studies even though your drug is not double negative. And also in the planned stage 2, we are going to develop Q&As to discuss how these proarrhythmia models or algorithms can be used to inform clinical decision-making for QT prolongers. Even though those algorithms are not written into Q&A yet, the follow-up studies can be used as of now on the case-by-case scenario for drugs that are not double negative. Thank you.
Thank you very much, Ziwa. The next question going to direct to Jean-Pierre Valentine from FPIA. And the question gets at that, the question was stated, this implies that non-clinical studies for QT risk are required for oncology drugs. Jean-Pierre, can you comment on how the new S7B Q&As serve as an option and for oncology drugs and the relation to the ICH S9 guideline for non-clinical evaluation of oncology drugs?
Yes, clearly when a TQT or TQT substitute is not possible or feasible, such as in the case of oncology drug, a sponsor may decide to follow the ICH, the non-clinical ICH S9 guideline, acknowledging upfront that the non-clinical data will have limited, if any, impact on the regulatory decision making and the overall non-clinical to clinical integrated risk assessment. What this new question and answer offers in terms of opportunity is offering opportunity to a sponsor to use high quality non-clinical data as described in this current set of question and answer that will be described in detail tomorrow, combined with phase 1 ECG data to form an integrated non-clinical to clinical risk assessment in order to impact the regulatory decision making.
Thank you, Jean-Pierre. There was a follow-up question on a similar topic based on Ziwa Li's presentation and discussion of the example of the oncology drug and what the sponsor would need to do. Would both clinical and non-clinical studies be needed? Ziwa, would you like to add any more details that builds on Jean-Pierre's response and the example you presented in your presentation? Ziwa, we're not hearing you if you are speaking. Okay, we're going to go on to another question. There was a question about using additional technologies, advances in non-clinical models to predict risk. I'd like to note that we will have a whole presentation tomorrow on proarrhythmia models and the set of Q&As in S7B about considerations for the use of proarrhythmia models. So I encourage everyone to tune in to that presentation tomorrow for more details. I am going to go to a question for Christine Garnett. Does the change to E14 Q&A 6.1 mean that oncology drugs are no longer judged based on the 20 millisecond threshold and how this relates to the 10 millisecond threshold that's mentioned in the Q&A 6.1 draft?
Yeah, this is a really good question. I think there's a lot of uncertainty about how to interpret the data coming from 6.1. So we step back, the original 6.1, we had a paragraph in the Q&A that talked about decision making. And so if a drug is being evaluated in an alternative study design, and because that study design does not have most likely does not have a placebo, it definitely doesn't have a positive control. And a lot of times it's studied, the doses studied in that alternative study design are about the therapeutic doses. They don't go higher. So we're really limited in the sensitivity of that study to detect small or to rule out small QT increases as stipulated in the E14 guideline. So the way we interpret the clinical data coming from such a study is that if the upper bound of the mean QT effect is less than 10, then we think that that data probably we're not missing a large signal as we like to define as greater than 20 milliseconds. So the idea is if the upper bound is less than 10, then we say there's no large mean effects. So now with the changes to the 6.1, hello? Anyway, so with the changes to the 6.1 guideline, to the 6.1 Q&A, what we're doing now is saying that if you don't have the non-clinical data or the non-clinical data is not conducted according to best practices and you don't have that, the extra sensitivity in the in vivo animal study, you would still be that criteria would still apply to the alternative clinical study data. However, if the sponsor does have that double negative non-clinical assessment, that we're going to rely on that assessment. And then if the QT data from the clinical study, the mean is less than 10, we're not going to be so much focused on that upper bound anymore, because we're now looking toward the non-clinical data to help fill that gap. I think together that would now be used as a low risk evaluation for QT. Again, as I answered earlier, that probably means that the company would not have to go into an expanded ECG program in phase 3. And then if there, again, at the time of marketing and application, if there's continues to be no signal in the adverse events that there is a proarrhythmic risk, then together that package would probably support I think from an FDA perspective, it would support a labeling language consistent with a no QT prolongation. Again, that's an FDA labeling recommendation, a labeling point of view. Other regulatory regions may have different perspectives on labeling.
Thank you, Christine. I'd like to ask you a couple additional questions. I'm going to mention them both. The first one is about high-quality ECGs and what exactly is meant by this. We had a number of questions come in related to this, and why don't we focus on that question, and then I'll ask you one additional after that.
Sure. Yeah, I saw a lot of people are asking about high-quality ECGs, and all my case examples, I said they had to have high-quality ECGs. So what I mean by that just is it's the same quality of ECGs that a company would use in their thorough QT study. So I'm looking for that they are in replicate. The ECG recordings are in replicate. They're taken in subjects who are, supine, at rest, and that I prefer that they're digital so they could be submitted to the ECG warehouse so we can evaluate the quality of the ECGs. That's what I call high quality. These are the same type of ECGs in a thorough QT study. I'd like to just take a moment just to contrast that to safety ECGs, and especially when it comes to 6.1, when companies are collecting ECGs, maybe especially in oncology trials, they have safety ECGs. They come on paper recordings. And they're not collected in replicate. Those, I would say, are not what we're looking for characterization of the QT effect. We're really looking for those replicate digital ECGs for characterization.
Christine, could you briefly comment on the question that came in was how many subjects are needed for QTC modeling, I believe referring to concentration QTC modeling and how to determine. Could you briefly comment on that?
Sure. So this really comes back to Q&A 5.1. And the working group, when we published that Q&A back in 2015, I started a working group to write a white paper. So we published the white paper that pretty much goes into talking about how to use data coming from a phase 1 study, how to model that to support 5.1. But to come to the question, so when you're thinking about how many subjects are needed in a phase 1 study to be adequate, it's not just the number of subjects, it's the number of subjects as well as the exposure range that will help with the power of detecting or excluding small effects in your phase 1 study. So a lot of times this study comes in with several dose cohorts. Maybe there might be 4 or 5 dose cohorts. In each dose cohort there may be like 6 subjects on drug and 3 on placebo, and so the number of subjects per se is not important, but it's per each dose cohort. But we want to make sure those dose cohorts have a large exposure range, and so it's the combination of the two. If the high clinical exposure dose is the highest dose in that study, it's probably not going to be sufficient. We really want to make sure that we have that 2x margin to be able to exclude those small effects. And that's, and if you want all the details for that, we did put that into the scientific white paper publication.
Thank you, Christine. The next question I'm going to ask Colette Stradanova from Health Canada to respond to. I know Colette was having some mic issues earlier, but I think they're resolved. And if not, I'd like to ask then Christine to respond to this question. So in the E14 Q&A 6.1, it discusses how a totality of evidence approach is taken, and that involves the non-clinical studies that we've discussed, having double negative, and that you would have high-quality ECGs in the alternative clinical QT clinical assessment that doesn't suggest QT prolongation. That is number 2. And then the third item is a cardiovascular safety database that does not suggest increased rate of adverse events that signal potential for proarrhythmic effects. Colette, could you comment on that number 3 criteria a little bit more?
Yes. Regarding the cardiac safety database across studies, the assessment would include examining adverse events suggestive of proarrhythmia, such as palpitations, syncope, seizures, cardiac arrest, ventricular tachycardia, Torsade de Pointe, ventricular fibrillation of flutter, and sudden death, checking for imbalances between treatment arms or dose groups. This can be challenging for single-arm trials. Causality assessments would involve considerations such as temporal relationship, underlying risk factors, and concomitant medications.
Okay, thank you very much, Colette. I'm going to go on to ask Ziwa Lee the next question. The question was about what and how many reference drugs should be used when determining the HERG safety margin. Ziwa, can you comment on the reference drugs?
Sure. The Q&A states that the reference drugs should have known clinical TDP risk and cover diverse electrophysiological properties. That means this list of drugs should include drugs that block HERG with different potencies, especially moderate and weak HERG blockers, because these drugs tend to be around the boundary of the safety margin threshold. On the other hand, the Q&A is not prescriptive and does not specify or support a any particular list. The sponsor could consider using a subset from some widely recognized lists of reference drugs, for example, CIPA list, JIXA list, Kudomet list. However, a justification should be provided in selecting subsets from these lists. Thank you.
Thank you. I have a follow-up question for you, Ziwa. In calculating the safety margin, what Cmax should be used for the reference drugs and for a new drug that you're evaluating the risk for?
For both the reference drugs and the new drug, we recommend the Cmax should be the high clinical exposure scenario, which are described in the presentations multiple times. Specifically, the high clinical exposure is the steady state maximum plasma concentration when the maximum therapeutic dose is taken with intrinsic or extrinsic factors. This is the exposure that the supratherapeutic dose in a thorough QT study is supposed to cover or exceed. Thank you.
Thank you, Ziwa. So we still have multiple additional questions that we're going to address this morning, or today, I'll say, before we end. But I wanted to briefly have Derek Leishman from Pharma comment on we have seen a number of questions come in that relate more to tomorrow's topics, and Derek will be facilitating the Q&A, the live Q&A discussion tomorrow. Derek, could you briefly summarize some of the questions that have come in that we'll be able to address in presentations and the live Q&A tomorrow?
Yeah, sure. Thanks, David. So we have been monitoring the questions in the background, and we have, owing to the number of questions, we have been having to triage them to try and get the questions most relevant to today's presentations to be addressed. But there have been a number of questions that have come in. The particular ones that people are interested in, of course, are how we set the sensitivity for the in vivo study, and that will be covered in some depth tomorrow. And also some further details on the ion channel studies that we've been hearing about, the HERG channel and so on. Dr. Lee gave us some insight into the margin calculations that will obviously be relevant to the ion channel work tomorrow too. The other piece that will be of interest tomorrow, we've had some questions relating to looking at beyond QTC and to proarrhythmia. As you can see from the Q&As, that's really a stage 2 activity, but we do have a couple of Q&As tomorrow which relate to the proarrhythmia assessment. So we will be looking at cardiomyocytes tomorrow and the principles of proarrhythmia assays, so that should set us well up. So the questions have come in, we will be addressing them, so just reassure the audience that they'll hear more tomorrow.
Thank you, Derek. So there was a question that came in about the global regulatory agreement on some of the content, and I wanted to highlight, as I mentioned in the introductory presentation, these Q&As, which are currently in step 3 of the ICH process. And the step 3 is where we are consulting the public, and there are official comment periods open in the different regulatory regions where anyone can submit official comments. That is encouraged. And what will happen is the working group will then, the ICH E14 S7B working group, discuss those comments, and then we have the ability to make revisions and then finalize. But the Q&As that have been published by ICH as the draft Q&As, they have been developed by the entire working group that includes representatives from about 7 or 8 different global regulatory agencies, and also representatives of each of the 3 industry groups: pharma in North America, FPIA in Europe, and JPMA in Japan. And the entire working group came to consensus on the draft Q&As, and the regulatory regions have adopted the draft guideline, and that is what has happened to date. We're gonna try and get through a few more questions. We still had many more that came in. I would like to direct the next question to Ziwa Lee. There was a question about comparing the safety margins in the in vitro HERG assessment. The question is, what is the point of having an estimate higher than the reference drugs, but the confidence intervals are overlapping together? Can you comment on this, how the confidence intervals of the estimates come into play, Ziwa?
Sure. Yes, from a scientific point of view, when we say a drug is above a certain margin, we mean the lower end of the confidence interval is above the region. above the threshold. If a drug has its medium above the threshold, but above the threshold, but the lower end of the confidence interval is not, then this is one example of the borderline HERG positive drug I mentioned in the third example. So if a drug is borderline positive, then by definition it is not double negative. However, you can use follow-up studies to further prove that your drug is actually of lower risk. Thank you.
Thank you, Ziwa. I have another question for you. Is there a need to make a correction with protein binding for the in vitro HERG assay?
Yes, as stated in the Q&A, by Cmax we mean the free Cmax or unbound fraction of the drug in the plasma. However, in some rare situations when you believe that the tissue accumulation or the protein binding fraction cannot be accurately assessed, then you may use the total Cmax of the exposure. But for most cases, we expect to see the use of unbound fraction, which means you correct for protein binding. Thank you.
Thank you, Ziwa. The next question I'd like to ask Jean-Pierre Valentine from FPIA. What about drugs that are actually in clinical development, currently phase 1 and beyond? And what do we do about them, and how do the new Q&As potentially affect the drugs that are currently in clinical development?
Thanks, David. It is effectively an important question. It is acknowledged that there will be a transition period during which drugs that are actually in clinical development would not have been through the newly described and proposed standard for the non-clinical assay. That said, if the sponsor goal is to support a standard TQT path, then the non-clinical core battery assay as defined in the original ICH E14 may be appropriate and sufficient and adequate. However, if the sponsor goal is to support question and answers 5.1 and 6.1 or 6.1, then the non-clinical core battery assay may need to fulfill the proposed best practice as defined in the new question and answers that will be presented in greater detail tomorrow.
Thank you, Jean-Pierre. I have an additional question for you. Beyond the ICH S7B Q&As compliant assays, a sponsor may have additional early discovery non-clinical safety studies to support claims about the safety of a drug. Would these early discovery safety studies be considered as a part of the non-clinical-clinical integrated risk assessment?
Again, an important question, and it is also acknowledged and recognized that sponsor may have at their disposal a number of non-clinical safety data regarding the drug's potential to affect either cardiac repolarization, QTSA prolongation, or the TDP risk. Such non-clinical data result may be used to support a claim about the safety of the drug. However, the weight carried by such study in the overall non-clinical to clinical integrated risk assessment will largely depend upon the ability by the sponsor to demonstrate the quality, the robustness, and the confidence in the study result, and consequently their possibility to be used to interpret and contextualize the results.
Thank you, Jean-Pierre. We have a couple questions that get back to clinical exposure response QT modeling, and I would like some combination of Christine Garnett and Charles Benson to address these. How about Charles for this first one does the statement that exposure response modeling should rule out a 10 millisecond QT increase and suggest no relationship between concentration and QT mean that the mean regression line must have a flat or negative slope? And what about what if there is a positive slope?
Right, yeah. So whenever you do a regression, a linear regression, you're either going to have a negative or positive slope. You're never going to have a slope which is absolutely zero. And so then perhaps the question was whether you have a clinically significant slope, and the answer is that what you need to do is look for ruling out 10 milliseconds regardless of slope at the relevant concentration. So in the new 5.1 is at the high clinical exposure. At the old 5.1, it's twofold the high clinical exposure. All of this is detailed in the Garnett white paper in the Journal of Pharmacokinetics and Pharmacodynamics, June 2018. So take a look at that.
Okay, Charles, I'll ask you an additional question. So the presentations made reference to obtaining concentration QT data specifically in phase 1. The objective is to have QT data across a large range of concentrations. This could be in phase 2 or phase 3 studies where concentrations are obtained over time from the Cmax down to lower concentrations. And could you comment on this and whether additional data can be used beyond phase 1?
Sure, yeah. And so the answer to the question is yes, you can obtain additional QT data outside of phase 1. However, phase 1 is especially suited to obtaining the information that we're looking for, especially in regards to the concentration response, because typically you're doing intensive pharmacokinetics during phase 1, and you could obtain your triplicate ECGs at the same time you're obtaining your pharmacokinetic data to facilitate the concentration response modeling. Also, phase 1 units are especially good at obtaining QT information, much more so than the more outpatient-based phase 2 or phase 3 studies. And finally, and perhaps most importantly, that phase 2 and 3, your visits are much more spaced out. And so, again, looking at that white paper, you'll want to obtain baseline ECGs prior to getting drug, and then obtain your QT measurements reasonably soon after. If you wait a number of weeks after, comparing back to baseline adds variability, and we try to decrease variability as much as possible in order to, again, rule out these very small changes.
Thank you, Charles. Christine Garnett, could you comment a little bit more on how we consider the intrinsic and extrinsic factors and whether you're looking for the one with the greatest increase or you consider a combination? Are you able to elaborate on that?
Sure. So we typically look at the worst case scenario, which is a lot of times we consider only one of them. We don't require or we don't ask companies to combine different intrinsic and extrinsic factors to come up with, something larger. So we take more of a simplistic approach and just say, what is the worst case in terms of what is the intrinsic or intrinsic factor that increases exposures the most, and that's where we will what we will consider. Thanks.
Okay, thank you very much. We are coming to the end. I would like to thank all of the presenters. I would like to thank the additional working group members that joined us for the live Q&A. And I would like to thank the entire ICH E14 S7B Working Group for all of the work over the past 2 years to develop these Q&As and get them out in draft form, and then to prepare for this webinar. I encourage you to make comments through the official comment periods if you are interested. And tomorrow we have many more things to cover. Derek Leishman will give a from pharma will give an introduction to the day 2, and we will subsequently cover in day 2 more detail about the S7B Q&As, the best practices for the in vitro assays that covers ion channels and additional myocyte studies. We will then cover best practices for the in vivo studies and the principles of proarrhythmia models, and have another live Q&A discussion. So with that, I would like to turn it over to Ray to make some closing remarks.